In this video, I'm going to go ahead and try and make a connection between the anatomy of the eye and the nervous system. In other words, how do the light receptors convert light images into an electrical signal that can be sent to the brain? And this, of course, happens in the retina. And so the retina is in the back of the eye here. And what happens is that as light comes in and hits the retina in all its areas here, it's going to be absorbed by photoreceptors and then converted into an electrical signal. We call that transduction. And so if you take the retina here and you look at it really closely, a little section of it kind of looks like this, and it's filled with a bunch of different nerve cells there, and we'll go over all of them. And eventually it's going to go down, of course, the optic nerve, which will send it off to the visual center of the brain, which is in the back occipital lobe. So we're going to talk about this today, what's going on with all these nerves here that are going to capture light and convert it into an electrical signal. So let's take a close look at the retina. So here's a little slice of the retina, and just in case you're lost, this picture is going this way. So light is coming in from the front of the eye, right? And the interesting thing is that the light goes all the way through the retina layer till it hits the back, the very deepest part of it, and that is the pigmented cells. Okay? So let's go ahead and call that the pigmented cells. And that was that colored portion we did in the dissection. Okay? And attached to the pigment cells are the two types of photoreceptors. We have two types. They are called cones and they are called rods. So these are the photoreceptors, right? You can see that they look kind of different. The cones conical, have a little peak like this, and the rods are just rounded off. Rods for rounded, cones for conical. Okay? So light then gets bounced off, right? Bounced off of the pigments, and it then goes back to the front of the eye. Okay? So it's going this direction. These guys, the purpose of the rods and cones is to take that light and convert it into a chemical and then an electrical signal. So the way they do it is start off here, and then we go to the next set of cells here. And the next set of cells here are called bipolar neurons. Now the reason why they're called bipolar neurons is because here is the nucleus, and you can see they've got two little pieces like that. So, I'll kind of draw it like this. So bipolar means they have two pieces coming off the nucleus. So the signal then gets chemically passed over to these guys, right? So then it goes this way. And these guys are going to convert the chemical signal into an electrical signal. And that goes then to the next set of cells here. And the next set of cells are called ganglion cells. So let's review it again. Light comes in all the way through the whole retina, bounces off the pigment, then goes to the conical cells, then to the bipolar cells, and then to the ganglion cells. And the ganglion cells bring it all together. So they're going to bring the signals all together, right? And they bring it together into a nerve fiber, which we call the optic nerve, right? So the optic nerve, and then the optic nerve is going to go ahead and send the signal to the brain. So light, pigments, cone and rods, bipolar, ganglion, and then optic nerve. Okay? Let's go ahead and erase this for a second, and then we'll talk about it in a little more detail here. Let's go ahead, if I can just erase this. Yeah, good. Okay. So let's take a look at it in a little more detail. Okay, here's the pigmented layer, and you can see the rods and cones that are there. And the thing you notice is that all of the rods look the same, right? There's a rod, there's a rod, there's a rod, but you can see that there's actually three different types of cones, right? Okay. There's a red one, there's a blue one, and there's a green one. So what's the deal here? Well, let's take a look. So let's make a tea table here. And not a very good tea table. Let's go ahead and talk about the differences between rods and cones. So the first thing is that rods are more sensitive to light. So they can really tell the difference between a lot of light and a little light. Cones are less sensitive. Why is that? Because rods are basically for black and white or gray, and they're basically used, therefore, at night. Whereas cones are used for color, so they're better in the daytime. Rods are our black and white vision. Cones are our color vision. As we saw in the picture, all the rods look the same, but there are red, green, and blue pigments in the cones. Okay, so don't say that they're red, green, and blue cones. They're not different colors. They have different pigments that absorb the wavelengths of red, green, and blue light. Now here's another thing. So we said that the photoreceptors then pass the signal to the bipolar neurons. The rods pass it from many rods to a single bipolar, whereas the cones go from one cone to a single bipolar. So let's take a look, for example. Go back to the picture here. So here, one cone is connected to one bipolar. However, with the rods in this picture, it could be many. So I see 3 rods here are connected to one bipolar. What's the difference between that? Well, it means that you're going to get a more exact reading of the light and the color if it's a 1-to-1 than you would get in the darkness as you go from 3 to 1. So this system here is going to be more exact. We have a word for that. We call it acuity. Acuity means how sharp the image is. So because many rods are connected to one bipolar, we don't have a lot of sharpness acuity, but because in cones, one is connected to one bipolar, it's a very sharp image. We have a lot of acuity there. So that's the difference between the rods and the cones. Now, what's going on in the cone pigments there? So I said that there were pigments that absorb red, those that absorb blue, and those that absorb green. These pigments are called opsins. Okay? The gene for it is located on the X chromosome, right? So you could have an allele for red, you could have an allele for green, you could have an allele for blue. And depending if you're deficient in them, that would cause color blindness, which is typically red-green color blindness. Now, let's take a look back at the picture here for a second. I'm going to go ahead and erase this. And you find that the distribution of rods and cones is not very equal. So where are most of the cones found? Most of the cones are found in the middle area of the retina. So they're going to be found, I'm going to draw this a little better than that. So most highest density would be found like there. Where is the absolute highest density? It's right in this little area right there, yes? That's going to be your highest concentration of cones where you have the best color vision. And that area is called the fovea centralis. Okay? Let's see if I can fix that up so you can see it. So the area where you have the most cones, no, still not working for me, is going to be, there we go, the fovea centralis. Okay, excellent. Where are the most rods found? Well, it's interesting that most of the rods are found at the periphery of the retina. So over here and here. So this is why when our peripheral vision, like if you're just walking along and all of a sudden something catches your attention on the right or left, you see the image, but you don't have a good sense of what the color is, right? Now, what would happen in animals that don't have a lot of cones? They would have very, very bad color vision. For example, dogs, they don't have a lot of cones, so they see things mostly in black and white. Okay, let's go ahead now and talk about one last thing that we'll finish up on. The octopus and the squid eye. Let's go back and look at our picture one more time, and you'll see that there's something really weird about this situation. And the problem is it's backwards. Okay? So let's review. So just so we know. Here's the front of the eye, right? That's the front. And here's the back. Yeah? Okay. So let's review what's going on. Light is coming in this way, goes through the whole retina, then it comes back through the rods and cones, goes to the bipolar neuron, goes to the ganglia, goes to the optic nerve. Then the optic nerve in the blind spot goes back all the way and then goes to the brain. Weird, huh? In, out, then in again. And that's why we have a blind spot here, because the optic nerve needs to get back in through the retina so it can go to the brain. However, there are some animals that have come up with a better solution, the octopus and the squid. So here's what we do, right? We come in like this, then we come out to the optic nerve, and then this guy goes in here. Not so for the octopus and the squid. For them, their rods and cones are backwards. In other words, not really backwards, they're actually in the right order. So light comes in here, gets absorbed by the rods and cones, goes to the bipolar, goes to the ganglion, goes to the optic nerve, and then it goes to the brain. That's the way it should be. In other words, there is no blind spot here like we would have in our eyes.